جامعة كيوتو. Shall I speak this introduction in Arabic only? It's okay? Arabic. Okay. جامعة كيوتو عمرها حوالي أكثر من 115 سنة. الجامعة حصل أعضاء هيئة التدريس فيها حوالي 9 أعضاء هيئة تدريس أو باحثين، 4 منهم حاصلين على 4 جوائز نوبل في علم الفيزياء، 2 في علم الكيمياء، 1 أو 2 أعتقد على ما أتذكر في العلوم الطبية الإنسانية. بالإضافة إلى كده إن هم بدأوا في المرحلة الأخيرة يعملوا كورسات بوست جرادويت ماستر بي اتش دي في اللغة الإنجليزية بغرض إن هو يجذب الطلاب الانترناشونال. يعني طبعا هو لازم يكون ليه كوتا معينة من عدد انترناشونال ستودنتس على أساس ياخد هاي رانك. هم ترتيبهم على مدار ال5 سنوات الماضية ثابت 22 انترناشونال رانكينج من 22 ل 23. اللي مش مخليهم يطلعوا في ال10 أوائل هو عدد الطلاب الانترناشونال. ده كلام اللي اتعرض من الفايس بريزيدنت بتاع الجامعة. ده الكامبس بتاع جامعة كيوتو. لو في أي استفسار بعد ما يخلص المحاضرة بتاعته يعني سواء باللغة العربية أو اللغة الإنجليزية، لكن مش باللغة اليابانية طبعا. أقدم لكم بردك بروفيسور سوكاوا. هو تخصصه بلانت بايوتكنولوجي أو التكنولوجيا الحيوية النباتية. التكنولوجيا الحيوية النباتية، وبيدرس حوالي 5 كورسات خلال العام الأكاديمي. حاصل في عام 2005 و2006 على جائزة المجلة اليابانية لعلوم البستنة، وأيضا على جائزة الجمعية اليابانية لعلوم البستنة في عام 2006. في الحقيقة هو زيارته ليست فقط كزيارة تبادل أعضاء تدريس أو أعضاء بحوث، هو زيارته تفعيل أول خطوة في اتفاقية. معظمكم سمع عن الاتفاقية اللي تمت بين كليتنا، كلية العلوم والدراسات الإنسانية جامعة سلمان بن عبدالعزيز وجامعة كيوتو. فمن ضمن الايتمز في الاتفاقية ستاف اند فاكولتي اكسشينج والستودنت اكسشينج. ولسه امبارح في المناقشة معايا عرض إن هو عايزين يبعتوا الطلاب من جامعة كيوتو للكلية عندنا. والحمد لله امبارح كان عندنا اجتماع مصري مع زملائنا الافاضل رؤساء الاقسام المختلفه بحيث تفعيل التعاون ايضا في المجالات المختلفه سواء الفيزياء، الكيمياء، اللغه الانجليزيه، معظم التخصصات عندنا في الكليه. ده تقريبا كيمستري فيزيكس. انت عرفت ده 2008 بردك حاصل على جائزه نوبل. دول الحاصلين على جائزه نوبل خلال من عام 1949. أول جائزة نوبل حصلوا عليها كانت عام 1949، يعني almost 70 سنة تقريبا. طيب أنا مش هطول أكتر من كده بالنسبة للانترودكشن. الفيديو ده ممكن أخليكم لو زرتوا بس كده على جوجل كيوتو يونيفرسيتي هيطلع لكم كل المعلومات، الموقع باللغة الإنجليزية، باللغة اليابانية، باللغة الكورية، واعتقد ايضا باللغه الصينيه. حاطين كل المعلومات افيلابل، الفيديو، الاقسام والتخصصات المختلفه. اعتقد كده اخلي البروفيسور سوكاوا يبتدي المحاضره بتاعته. هي هتبقى عن تقنيه انتاج نباتات خاليه من الفيروس والفيرويد باستخدام تقنيه زراعه الخلايا والانسجه النباتيه. طبعا الفيروس والفيرويد بيعملوا مشاكل كبيره جدا في انتاج النباتات وخاصه اكسارها. I will introduce Professor Sukawa. Please, the floor is for you, please.
Thank you for introducing me. At first, today, I must say special thanks to the Dean and Vice Dean and Professor Sharaf Eldin for visiting me and giving me a chance to have a presentation about my works. I'm working about plant breeding using plant tissue culture for 20 years. In this time, I could have a chance to study about saffron in vitro culture. This is my first experience, and I have already challenged saffron in vitro culture in this several months. And I found saffron is very interesting crops, and it's beneficial for us and in my laboratory. So, I'm looking forward to have a collaboration with Professor Sharp. Yes, today I will have a presentation about my little old works. My English pronunciation is not so good and very hard to hear. So, I'm sorry for my slow speaking and read my original papers. I'm sorry. Okay, I will start. At first, I will explain about the difference between shoot tip and shoot apical meristems. Shoot tips is the top of the shoot in plants. This is the shoot tips. This is stem and leaves and small leaves. And when all leaves are removed, at last, this kind very small cells composing dome-like structure is observed. So, usually, plant researchers called this is shoot tips, and this is meristems. This is shoot tip, and this is meristem. Usually, we use shoot tips for horticultural use because shoot tips has no virus and viroid, no pathogens. But nowadays, many pathogens are found that can invade to the shoot tips. This is very small leaf, and this is undifferentiated cells. We call this small leaf is leaf primordia. This is the symbol tree of our university, cinnamon tree. And this is a seedling of cinnamon tree. This big tree is originated from only one meristem. So, meristem supplies almost all the cells above the ground. For example, this is a big cinnamon tree, and this is a strawberry plants in my university field. Horticulturally, the propagation of plant is usually dividing the meristems. We call this meristem is runner, and strawberry plants is propagated by runner. These runners is from one meristem, one terminal meristems. So these plants are clones. In horticulture, clone propagation is dividing meristems. this is in vitro culture for virus and viral-free plant productions. And this is shoe tips. Background of first study I introduced. Shoe tip culture is used for micropropagation and virus-free plant production. Because virus titers are reported to be lower at the top of the plants. Smaller pieces of shoot tips are better for virus-free plant production. But smaller is difficult to survive on hormone-free medium. Usually we use a plant growth regulator called plant hormones. And it helps the grow up and survive on the medium. However, there are many viruses and viroids which are difficult to be eliminated from infected plants by shoot tip culture. So, development leaf primordial free shoot apical meristem, and we call some shoot apical meristem, and leaf primordial LP free some culture without plant hormones is needed. And this is my process of my method. As mentioned before, shoot tip culture is normally used for virus-free plant production. This is shoot tip, top of the shoot, and beside the shoot tip and the culture. And this is usual shoot tip culture for virus-free plant productions. But there are many pathogens which are difficult to be eliminated by the usual method. So, because of the titer of the pathogens, usually decreasing acropetally, decreasing the top of the shoot. This is meristem. The lowest concentration is very top of the shoot tips, meristem. So, it is better to use the only meristem for free plant production. Only leaf primordial free shoot apical meristem is suitable for pathogen-free plant production. But usually only the LP free some is cultured on the medium, on hormone-free medium. It's dry up easily. So I tried several tissues or organs for rescuing leaf primordial shoot apical meristem, and at last we could find out the suitable organ, root tips. That is, if transferred the leaf primordial shoot apical meristem to the root tip, making artificial embryo, and it can grow up to the plants. Yes, because root tip has high activity of cell division and low content of polyphenols. It's a very good point for root tip for rescuing leaf primordial shoot apical meristems. This is a photograph for my method. This is a shoot tip, usually used for the virus-free plant production. But my method is using only the top of the shoot tips, meristem, and transfer to the root tips and making the artificial embryo and grow up to the plants. I think this is a very good method for the eliminating all of the pathogens from plants. you can support picking up leaf primordial shoot apical meristem is very difficult and laborious work. So my student cannot do the same works. So I developed apparatus for taking some easy. This apparatus. We put the shoot here, and this shoot has no leaf primordials. All leaf primordia were removed under the microscope. So put into in here, and this is laser. And adjust the position of the laser and slowly move the laser to the shoot apical meristem. This photograph is taken under the microscope. And finally, we can take very small shoot apical meristems using this machine. And this is one more apparatus for LP Frisson culture. This machine is a very simple one, using for the removing all of the leaf primordials. Leaf primordial removing is very laborious. We must conduct all the work under the microscope, but it takes 2 or 3 minutes for one plant. So it's very laborious for student. So this is compressor, easy compressor. We can buy at the home center. And this is a additional tanks for compressed air. And this too is sterilized air. And this is for the dryer. And this is the final filter for sterilizing the compressed air and air gun. And if using this air gun, we can remove all the leaf primordia within 2 or 3 seconds, and the result microscope. It's very easy. So if a student can use this machine and remove all the leaf primordia, I can put the shoot. And this is a photograph after the culture. This one is 7 days after leaf primordia shooting culture. This is a root tips, and this is a transferred leaf primordia shoot apical meristems. A new leaf primordia is generated from LPP cells. And this is 30 days after culture. Visible leaves can be observed. And vascular bundle is connected to the root and shoot. This is a result of this table shows the percentage of the success of leaf primordial shoot apical meristem culture. When the shoot apical meristem are cultured without root tips, this one, no root tip. This is a cultivar of the chrysanthemum plants. Chrysanthemum is a very famous ornamental plant in Japan used for ceremony. And no root tips, no success. But when the chrysanthemum shoot tip is LP free shoot apical meristem is cultured on the root tip of chrysanthemum, high percentage of success was achieved. Yes, this one. When even the different cultivar, first cultivar and second cultivar, high percentage of success was achieved. Very good result, I think. But this is a very big problem. LP-free sum culture using chrysanthemum root tips. The photograph of the connection of the vascular boundary is using the chrysanthemum root tips and chrysanthemum shoot apical meristem and chrysanthemum root tip connected. But chrysanthemum root tips is difficult to use for viroid-free plant production. Later, I will introduce explain about the viroid. Yes, this is a very small pathogens, a big problem in Japan. Because almost all chrysanthemum cultivars have been infected by viroids. Viroid diseases are most dangerous ones in the world, and these infectious agents are most difficult ones for eliminating by usual method, usual shoot tip culture. We had to search out a new plant species for root tips. Viroid is a very big problem in the Mediterranean areas. For example, for peach and tomato and potato. Also Japan, chrysanthemum. Also in Japan, chrysanthemum viroid is very big problem. And I screened suitable plant families among over 20 species for chrysanthemum, for rescue the chrysanthemum leaf primordial shoot apical meristems. For example, cabbage, petunia, dianthus, carnation, and chrysanthemum. It's a normal method. No root tips. No root tips can survive the shoot apical meristem, but petunia and cabbage plants can survive, can rescue the shoot apical meristem of chrysanthemum plants. And especially the cabbage root tip is very effective for surviving the shoot apical meristems. Out of 20 plant species belonging to different plant families, plants belonging to brassica, cabbage belonging to the brassica family, were revealed to good materials for rescuing chrysanthemum sons. And I used several cultivars, Piato, Iwano Haxen, and Statesman. These cultivars are main cultivars for chrysanthemum crops in Japan. So all cultivars, for all cultivars, cabbage root tips is very available. So high percentage of viable plants obtained. And this photograph is very interesting one. This is a shoot apical meristem of chrysanthemum. This is a root tip of cabbage. The cell-to-cell connection was obtained at 7 days after some cultures. Cabbage and chrysanthemum is thoroughly different families, but thoroughly different families, also thoroughly different families, these two organs can connect temporarily. Cells between two organs of different plant families are attached at 7 days after culture. Yes, but these two organs separate additional 5 days culture. So maybe chrysanthemum should tips know the cabbage is different families, additional 5 days. In this stage, LP-free sum can arrive without root tips. So separating the root tips and the shoot is no problem after the 12 days culture. This temporal cell-to-cell connection is very important for surviving the shoot apical meriscems without plant hormones. And I must introduce about the viroid. Viroid is the smallest known pathogen without protein coding regions. Thank you. This pathogen is composed of RNA, only RNA, without DNA and without proteins. And no coding regions for proteins. Transmission, viral transmission is achieved by cuttings, vegetative propagation in horticulture. For example, crop production from one infected potato propagated by human, infection is spread to the next plant generations. So, and once infected plant observed, we must dispose them. No way, there is no way to clean up the virus once infected plants. Once infected plants, we must find out and dispose. And viroids can classify to two groups, PSTVD group and ASPVD group. PSTVD means the potted spindle tuber viroid. This viroid is the type viroid in this group. And ASPVD group, this viroid is avocat sunroach viroid, ASPVD. And chrysanthemum stand viroid and chrysanthemum chlorotic motor viroid. And these two viroids is very big problem in Japanese chrysanthemum productions. And no way to clean up the infected plants. And these two group is PSTVD group viroids can proliferate in nucleus and exist nucleus. Sometimes go out the nucleus and cytoplasms for transmit to the next cells. And ASPVD group viroid is proliferating chloroplast. Both types are big problem in the world. In Mediterranean areas, each viroid is this type. And this is a symptom of chrysanthemum chlorotic motor viroid. This is a chloroplast type. Discoloration of young leaves and stunting is a main symptom of these pathogens. This kind of discolorations. This one is more obvious. This is a symptom of the chrysanthemum stunt viroid. This is nucleus type. Most big symptom is stunting. So I would like to clean up these infected plants by using LP-free some cultures. Yes. And this photograph is in situ hybridizations. The black spot. Each black spot indicates a viral infection. So this is a meristem. This is a transverse section of the infected plants. This is a meristem and young leaf primordial, and developed leaf. This is a free area. CSVD, chrysanthemum stand viroid, is not infected in this area, but the black spot is infected. So this photograph indicates CSVD can embed to the top of the shoot tips. So we must pick up this small area and cultures, and maybe we can get a free plant using LP-free some cultures. This is a LP-free some culture. Root tips and shoot apical meristem is combined, and cultures and individual plants getting. And again, we put the shoot apical meristem from the individual culture and root tips, and we can repeat this cycle, and perfect free plant production is achieved. Yes, this is a result of the LP-free some culture for viroid-free plants. This table means the CSVD-free plant productions by LP-free some cultures. The percentage of the free plant production was very low, 3%. But free plant could be obtained by this method. This is the first report for production of the viroid-free plant productions. And repeat of the LP-free some culture is effective, 16.7, but only one plant can obtain. I must ascertain this result. But in conclusion, we can get the CSVD-free plant from the infected plant. This is very important point. And next, I tried for making a free plant of CCHMVD. This is a chloroplastic type. Also to the CSVD plant, I can get CCHMVD plants, free plants from the infected plants by using LP-free some culture. The percentage of success is very low in this time also. But in this time, I can get the CCHMVD plant using my method, this one. And okay, and this photograph indicates the CSVD free plants. Okay. Yes, CSVD free plants grew vigorously, and an interesting phenomenon was observed. I will introduce this point. this one. This is a free plant generated by LP-free some culture. This is infected plants before LP-free some cultures. The free plants grow vigorously. Interesting point is this one. CSVD free plants could not flower under long day conditions. This infected plants can flower, but the free plants cannot be observed flowering. There is two types of chrysanthemum plants. One is day neutral plants and one is short day plants. And chrysanthemum in this cultivar, this cultivar is thought to be short day plants. Day neutral plants. So this cultivar can flower in the world whenever in the year. But if I conduct a LP-free some culture using this cultivar, I can observe the no flowering plants. Cutting from mother plants, its flower early August. This is a normal vegetative propagation, and this is a normal shoot tip cultures. And these plants can flower end of August, and LP-free summers end of September. This is one more photograph. This is a treatment of LP-free some culture. Sometimes flowering plants can be observed. And this is cutting plants. These plants could flower normally. And this is a normal shoot tip culture. Also, this plant can flower normally. But only the LP-free some cultures, plant cannot be flowered. And analysis by PCR for detection of CSVD infection to the plants. So these flowered plants obtained CSVD, and ordinary shoot tip cultures obtained having the CSVD, and mother plants have a CSVD. These plants can flower normally, but if I conduct a LP-free some culture for eliminating all of the CSVD from plants, these plants cannot be flowered normally. So this is a figure for if cutting plants vegetatively propagated by infected plants, this can flower, and LP containing from some usual shoot tip culture can flower. But making plants from LP-free some, it cannot be flower. It cannot flowers. So, in conclusion, this one, CSV disturbed periodic responses of chrysanthemum. Yes. Chrysanthemum can usually flower under short day conditions. But if CSV infect the short day conditions, short day type chrysanthemum cultivars, it makes chrysanthemum easily flowered whenever in the year. Conclusion. 1. LP-free some can degenerate to plants on cabbage root tips even in hormone-free medium. And cabbage root tip is available not only chrysanthemum but also potato, tomato, and Japanese taro, and many plants. Yes, very available. And virus difficult to eliminate pathogens could be eliminated from highly infected chrysanthemums. And CSVD disturbed the photoperiodic responses of chrysanthemum plants. Cabbage root tips could be used for various crops. Second experiment, very short introduction, very short introduction. At Aichi Prefecture in Japan, this is a very big, many farmers for chrysanthemum production. LP some good cultures, chrysanthemum plants are used for cultural production now. But the different problem is observed. Free plants are infected during the field propagation or cultivation. Viral resistant cultivars need to be screened, but no one could find out the resistant chrysanthemum cultivars. There is no resistant chrysanthemum cultivars in Japan and also in Netherlands. We tried to screen resistant cultivars among many commercial cultivars using LP-free some cultures. This is a little different use for LP-free some cultures. This is a photograph of the experimentally made bred resistant chrysanthemum plants at 2005. In resistant cultivars, CSVD resistant cultivars, CSVD disappears from the tip of the plant. This photograph is the result of this year. This is a usual infected plants. This plant have a CSVD all of the plants. All of the plants. But in the resistant cultivars, CSVD disappears from the top of the leaves. When infected plants used, I observed disappear. So I make this method for CSVD resistant cultivar screening. This is an illustration of the method. In this time, I used the CSVD infected root tips for screening. LP-free sum for determining the resistance or non-resistance. Cut from the shoot tips and transfer to the CSVD infected root tips. And after 2 or 3 months of cultures, this shoot tip has this meristem has CSVD or not have a CSVD. If this meristem, infected meristem, didn't have a CSVD, this plant supposed to be resistance. But if the infected meristem has CSVD, this plant supposed to be susceptible. And I checked a new method for simple method for determining the resistance of CSVD. This is only a pi as a very small bias and dip in the PCR liquid. So we can determine the infection of CSVD very easily. Yes. And we can screen two cultivars. First cultivars, commercial cultivars for CSVD resistance. This two cultivars, this one and one more this one. This one is a resistance, and this one is a resistance. This cultivar cannot proliferate CSVD in the plants, and this cultivar can decrease the CSVD concentration. And finally, this cultivar removes all the CSVD in the plant. I can find these two cultivars. Plants were infected with CSVD by grafting, and after 3 months, cutting were detached from root stocks. Data were collected from plants after cutting. This data indicates resistance on the CSVD. And once CSVD resistant cultivar was founded by our laboratories. So the Netherlands company and Japanese company screened more resistant cultivars. And now Japanese Sलमान company and the Netherlands Slमान company compete finding new cultivars. So this is a photograph of the normal cultivars and resistant cultivars. Same as a transfer section, and black spot indicates the CSVD. And resistant cultivar has no CSVD signals. Leaves sometimes CSVD signal is observed, but this signal cannot proliferate in leaf and shoot. Same. CSVD cannot transfer to the next cells. Okay. LP-free SAM culture could use for screening of resistant cultivars among a lot of commercial cultivars. Seino Issei, this is a cultivar name, Marikazakuruma, this is also the name. The first report on the CSVD resistant chrysanthemum. These two cultivars have different mechanisms for the resistance. One is cannot be proliferated in the plants, and one is decreasing the CSVD titers. And final studies. In Japan, there are several pathogen parthenocarpic tomato cultivars. Because these cultivars don't need pollination for fruit set. They are used for greenhouse tomato production in winter. In winter, very low temperature conditions, no insect is active. So the pollination is not succeeded. So parthenocarpic tomato cultivars can flowers and can fruit set autonomously. No pollination. So parthenocarpic cultivars should be propagated by cuttings because no pollination results in no seed. Decreasing plant vigor from plant aging because of the repeated cuttings occurred as new problems because of plant virus and plant vigors. We tried to develop a vegetative propagation system for parthenocarpic tomato using LP-free SAM culture for make the plants vigorous. This is a parthenocarpic tomato, NPK1. This is bred in our laboratories. we can see no seed. There is no seed in this tomato. And this plant can root set autonomously. But repeated cuttings make the plants grow bigger. And successfully, we can make a LP-free some culture also in the parcel-calpic tomato, and this method make this cultivar vigorous. So now, this is using several parcel-calpic tomato. Successful rate is very good. Rate of plant regeneration. And this is a field, tomatoes growers field. Now, in Kyoto area in Japan, it's not good conditions for vegetable production because of high temperature in summer and low temperature in winter. But this is a kind of a brand vegetables for growers can this tomato sell this tomato very expensive. So one of the brand cultivars. And one more example, cutting propagation for dahlia. This is one of the main crops in Japan. If we can make a free plants by LP some culture and making a big tubers. When making a big tubers, rain infection virus is a big problem. So I make a new method for cutting propagation of free plant dahlias. And also, I checked several viruses from dahlia plants. Many viruses and viroids was infected to the dahlia plants. And AP-free sound culture can successfully generate a virus-free plants. This is a result of the virus-free 68%, 80, 84, 44. Very high percentage of free plant production is achieved. And this is a result of the free plants, cumulative flowering cut flower productions. This is infected plants and free plants. Free plants can flower more production of cut flowers. Yes, finally. LP-free sound culture could be used not only for virus and viral elimination, but also the invigoration of vegetative propagated plants, for example, tomato. Okay. Thank you. That's all.
Thank you, Professor Scott, for the informative presentation. And if there's any question, okay, Sarah, please can ask.
Hello. Thank you very much for this talk. It's very nice. Although it's not my specialty, I'm a chemist, but I kind of understood many of what you said. Now, I have two questions. First, this virus, is it possible that it infects like Japanese fruits vegetables like cabbage, but not other countries, like not Saudi cabbage, for example? Is it possible that it's specific for Japanese, for Chinese, Korean, but not other? And in this case, is this study that useful, or can be extended? Now I have another question, which is it possible that this viral can go to humans, to animals, and transferred?
Okay.
I hope I was clear.
Okay, thank you for questions.
Excuse me, may I just translate it quickly in Arabic? السؤال هل هذا الفيرويد بيصيب فقط النباتات الموجودة في اليابان ولا ممكن يصيب النباتات الموجودة في السعودية؟ طب وإيه استفادة الدراسة دي في اليابان؟ السؤال الثاني، هل هذا الفيرويد بينتقل إلى الإنسان أم لا؟ Please answer.
Yes, thank you. At first questions. This method can be used for the many crops, and not the problem is not only Japan but the world. So this method can use the crops in your countries and in Europe and in USA. So this is available in the world. So and second questions. This viroid cannot infect the humans and animals and insects, but same kind of pathogens are found in animals nowadays, named hepatitis B virus. And that virus has no proteins, no DNA, only RNA, small pathogens. But viroid and that pathogen is completely separated ones. Thank you.
الإجابة إن الفيرويد بيصيب أي نبات في أي مكان في العالم وليس فقط في اليابان. السؤال الثاني هو لا يصيب الإنسان، لكنهم حديثا اكتشفوا إن فيروس الكبد الوبائي بي مشابه للتركيب الفيرويد الموجود في النبات، إن هو ليس ليه الغلاف البروتين. Thank you. If there is another question أو في أي سؤال تاني حتى لو باللغة العربية ممكن. So if there is no question,
thank you for giving me this chance. Let us suppose that we succeeded in producing plants which are free from viruses and viruses. Is there anything which guarantees that these plants, till the end of their life, will not be infected with outside with virus or viruses?
Virus can easily reinfect the free plants in the basically propagation of the free plants out of the in vitro. So we must keep the free plant in vitro, and propagation is conducted in vitro. It's preferred, I think. This is answer of your questions. Different.
Thank you. Is there anything which guarantees the continuation or till the end of the life of the plant being free will not be infected?
So you mean the particle transmission?
Yes.
Yeah. Okay. Viroid is infect to the seed. It's easily infected by pollen transmission. So if the plant is died in the life, but the viroid can transmit to the next generation. So it cannot be disappeared by infected plant will die.
I mean, for example, in greenhouse,
Mm-hmm.
if we have tomatoes free of virus, then for 2 months or 3 months they are free. But later they will be infected with virus and be stunted, and of course there is no remedy for the virus, and production will decline and till the end of the life. So in this virus-free produced plants, is there anything which is guaranteed that they will not be infected with virus till the end of their life? Would that be?
All is a plant which is resistant.
It is
resistant can be virus free or viral free, but if it is not resistant, it can easily be infected.
Yes, that's right. Yes, if the not free, not resistant plant, yes.
Thank you very much.
كان سؤال هل النباتات السليمة في احتمالية إن هي تفضل سليمة على طول غير مصابة بالفيرويد؟ الإجابة غير متوفرة، لكن لازم يبقى الإكسار دايما في نطاق تكنولوجيا زراعة الأنسجة أو زراعة نباتات عندها مقاومة إلى حد ما لهذا الفيروس.
I have a technical question regarding the size of the algal stream that you are choosing. Should be different from viroid and virus, and I would like to produce virus-free plants and viroid-free plants. How could you decide the size of the algal stream?
This is a very good question, I think. The size for the viroid-free plant should be very small, but for viruses, not so small should apical meristem is needed. Relatively large size is okay for virus-free plants. For virus-free plants, 0.3 millimeter, but for viroid-free plants, 0.15 millimeter is needed.
How about the success in tissue culture for such very small amount?
Yeah.
If you, for example, cultivate 100 explant for viroid free,
Yes.
how many plants should be might be survive?
Plant regeneration rate is
I mean surviving, because you are using very small amount of size of the explant.
surviving.
Surviving.
Okay. Now the surviving rate is 70 to 80%. It depends on the speed of the conducting. Yes. If my student conduct this method, the surviving rate is very low, but technically improved and the speed up the LP-free stem cultures, it can improve the surviving rate.
Thank you.
Welcome.
السؤال كان من زميلنا الدكتور مؤمن ايه حجم المقطع النباتي او الجزء النباتي اللي بينفصل من النبات على اساس احصل على النبات خالي من الفيرويد او الفيروس. كانت الاجابه الاثنين بيختلفوا، لو هتاخد النباتات عايز نباتات خاليه من الفيروس ممكن اكتر من 1.5 مللي، لكن لو عايز نباتات خاليه من الفيروس لازم المفصل او المنفصل النباتي اللي هو القبل الاستميه يكون حجمها ما يزيدش عن 1.5 مللي. نص مليمتر؟ أقل من نص مليمتر مش واحد، سوري، أقل من نص مليمتر. السؤال الثاني نسبة النجاح، أنت بتزرع مثلا 100 منفصل نباتي، نسبة النجاح بتبقى حوالي من 70 ل 80% والباقي ما بيستمرش كخلية أخرى أو نباتات. Is there any questions or أي سؤال تاني؟ Okay, if there's more questions, we would like to thank you, Professor Skar, once again for this informative lecture, and thanks for attending this lecture. Thank you very much.
Thank you.